Hi everyone. I'm just getting set up, and I think we'll get started maybe in a couple of minutes, just once people have time to log on. So yeah, if you just bear with me. Hi everyone. Hi Henry. Just admitting people into the meeting, and yeah, we'll get started very shortly. Maybe I'll just wait one more minute, just to make sure everyone who wants to be here is here. Hopefully you can see my screen. Let me know if not, but you should be able to see my slides. Brilliant. Thank you, Henry. Perfect. Thanks, Sandra. So I'll answer. We'll have some time for questions at the end. So I'll wait until the end to answer those. Feel free to put them in the Q&A chat. I know there's always a bit of confusion between the two chats, isn't there? So I will check both, but ideally in the Q&A would be great. And then we'll go through them at the end. So where are we? Shall I start? I think we might as well get started. So let me just navigate to my slides. So you should still be able to see them now. Right, so hi everyone. It's great to see you all here. For those of you that don't know me, I'm Rachel and I recently joined the company as Scientific and Technical Manager at Vesiculab. Before this, both my PhD and postdoc projects were EV projects, so I have an EV background myself. And if you have any technical questions after the webinar about any of our products, perhaps unrelated to the specific topic of today's webinar, do feel free to drop me a message anytime. My email address is going to be on pretty much all of the slides, so you can note it down or drop me a message on LinkedIn, whatever's easiest. And just a quick note to say we are recording the webinar, so we'll be able to share a link round after today, probably early next week. So just a brief intro to Vesiculab. We're a relatively young company, so we're based in the UK in Nottingham. We first started trading in 2023, and we sell various consumables and reagents that are aimed at researchers working on EVs or related lipid-based nanoparticles. And overall, we want to make extracellular vesicle workflows more efficient and actually work with you to get the best data out of your experiments. So this is by all means not an exhaustive list of what we offer, but I just wanted to highlight some of the solutions we've developed for different parts of the EV workflow, because today is focused on kind of one specific part, which is EV labeling. But we do have solutions for EV isolation. So we've got the Vesi-SEC system, which is a size exclusion based chromatography column formatted for centrifugation. So it's super fast and non-dilutive. I will talk about that a little bit later as well in the context of removing free dye. We also have Vesi-Safe, which is an EV preservation buffer. Let me get my laser pointer actually ready. And this is designed to preserve EVs long term in -80 storage, but also to prevent sticking to plastics. So it's better just for handling as well. to prevent particle loss. And then we've got our Vesi-Dye membrane dye range. So I'm going to talk a lot about this today, and so I'll skip over that. But then we've also got a rapidly growing portfolio of reference materials designed to help with EV standardization. And here I'm just showing two of the reference materials we offer. So on the left being a biological extracellular vesicle reference material that is engineered to be fluorescent, and is also actually suspended in that Vesi-safe buffer, so it's very nice and stable to work with. And then we've also more recently launched our Vesi-Ref rainbow beads, which are, as the name suggests, widely fluorescent across many channels, and are useful for tuning instruments for nanoparticle detection. So I actually gave an overview of everything that we're doing at the moment in my last webinar. So if you missed that, the recording is available on our website, or feel free to drop me an email and I can send you the link to that. So for today, I'm going to be just focusing on EV labeling with Vesi-Dye, and also how the workflow fits together with Vesi-SEC for free dye removal. Before we dive into EV labeling, I just wanted to take a step back and look at the overall EV workflow. EV research is challenging, and I know this firsthand, having done it myself. We're often working with precious, low volume samples. EV heterogeneity presents a huge challenge, and not only that, EV samples vary considerably between patients or experimental conditions, so every single step of handling matters. And throughout the workflow, there are several hurdles that we need to overcome, whether that's sample loss during purification steps, background signal from unbound dyes and antibodies, and I will be talking more about that in a bit, poor fluorescent signal or instability during storage. All of these can have a significant impact on the final quality and reproducibility of the data. So when you're working with fluorescently labelled EVs, these issues become even more important. For example, even if you have an excellent fluorescent dye, problems with residual free dye or inefficient EV isolation can make it difficult to then interpret your results with confidence. So at Vesiculab, our aim is essentially to simplify and improve the efficiency of EV workflows so that we can improve the sensitivity of EV detection and also reliability of EV data. So today I'm going to be focusing on EV labeling using Vesi-Dye, which is the range of lipophilic membrane dyes that we offer. And also I'll be presenting some published examples of downstream applications of the Vesi-Dye labelled EVs. So why do we want to label extracellular vesicles in the first place? Essentially, it allows us to discriminate EVs from non-EV particles. And fluorescent labelling has become an incredibly valuable tool for EV research because it enables both analytical characterization, but also it allows us to carry out functional studies. So from an analytical perspective, it allows us to identify and quantify EV populations, distinguishing them from total nanoparticles, and depending on the technique you're using, maybe doing a bit of multiplexing, so looking at multi-parametric analysis with multiple different fluorescent probes to do an in-depth sort of characterization. Whereas with functional studies, fluorescent labeling is essential really to measure EV uptake, biodistribution and in vivo tracking using techniques such as flow cytometry or microscopy, and it helps us to discern if an observed function is EV mediated. However, EVs do present some unique challenges compared to cells, so they're below the optical resolution limit of conventional light microscopy, and unlike cells, they don't express, unfortunately, a universal conserved marker. So we can't just label all EVs with a single antibody, for example. And so because of this, many researchers utilize membrane dyes as a universal labeling strategy for EVs. And membrane dyes, by intercalating into the lipid bilayer, they provide a simple way of fluorescently labeling the total EV population to then use across a wide range of downstream applications. So then it's a question of which membrane dye is best to use. Not all membrane dyes are created equally, and choosing the right dye with the right purification strategy is important for generating reliable data. And that's what I'm going to focus on for the rest of the talk. So some specific challenges that are important to consider when staining EVs. Many lipophilic dyes form micelles or aggregates, which can be a similar size to EVs and also be fluorescent, and therefore be detected as single particles, for example, by flow cytometry, which is what I'm showing here. And this is a real problem for false positive EV signals. So here we're showing the PKH26 dye mixed with buffer, no EVs in this sample, and you can see there's a significant population that's detected here, showing that it aggregates, and this could easily be confused with EVs essentially. Even without aggregates, excess unbound dye creates background fluorescence and can mask weak EV signals. And then also, to overcome this, researchers have to use cleanup protocols to remove that free dye. And they come with a load of their own pros and cons as well, including the cons being potential particle loss or sample dilution. And I'll talk a bit about that a bit more later. Another major problem with several lipophilic dyes is that they can undergo dye transfer between membranes, so that they can sort of non-specifically diffuse in and out of membranes. And this is a problem when thinking about uptake assays or biodistribution studies, where you're applying fluorescently labeled EVs, and when they come into contact with cells, the dye might just non-specifically transfer into the cell membrane, making it look like it's taken up an EV when it hasn't. So that can really impact the confidence that we have in the data. And finally, also problems with photo stability. Rapid quenching and photo bleaching can occur, leading to signal loss over time, and this can actually be very rapid, and that's particularly problematic for uptake studies over several hours or in vivo tracking over days. So ideally, we want a dye that is able to minimize aggregation, remain membrane associated and not diffuse freely in and out, have a bright and stable signal, and be compatible with many rapid purification workflows. So now I'm going to introduce Vesi-Dye. So Vesi-Dye permanently intercalates into the lipid bilayer. And you can see the schematic that shows that conventional dyes often don't span the entire membrane bilayer and are prone to diffusing in and out of the membrane. But the technology that was developed by Aquarela means that the Vesi dyes essentially permanently intercalate and span the entire bilayer. And they provide a very stable fluorescent signal with no transfer to cell membranes or any other bilayers. They're also water soluble. They don't form micelles, so they're reducing the risk there of false positive signals. And they're also fluorogenic dyes, so that means that the free dye doesn't fluoresce, and the probe only becomes fluorescent upon intercalation with a lipid membrane. And that means that it's got a really good signal to noise ratio. There's very low background noise when you label with Vesi-Dye. And the Vesi dyes come in a range of, they have a range of spectral properties available. So depending on your fluorescent setup, there's plenty of flexibility there when designing your experiments. So going back to some of the challenges with labeling, I'm going to just show how Vesi-Dye performs compared to other conventional dyes. So first plot here is showing essentially a flow cytometry experiment again. And here you can see that EVs are unlabeled here and measured. So you can see the EV population, but it's overlapping with background noise most likely. When we label with Vesi-Dye, this population emerges and separates out the positive EV signals from the background noise and any other non-EV particles present in the sample. The buffer simply mixed with the dye on its own and no EVs shows that there aren't any fluorescent particles being detected or any aggregates being detected. Whereas the buffer plus the pKh dye, you see that significant population emerge. So if you had an EV sample labelled with pKh, it would be impossible in this scenario to discern whether it was actually an EV or whether it was just a pKh aggregate. So this slide is essentially showing the same thing. So I appreciate it's really tiny and hard to see. So I've pulled out kind of the main figures that I want you to look at. And essentially, this is just another example comparing Vesi-Dye with DI8 on staining red blood cell derived EVs this time. And you can see that the Vesi-Dye mixed with buffer on its own with no EVs only gives about 2% false positive signal. That's kind of generally a threshold that flow cytometrists will use for isotype controls, for example. And then comparing that with DI8 mixed with buffer, see that significant population arise again, and that's because of aggregation and fluorescence of the aggregates being detected. And then you can also see the staining efficiency when the EVs and the dyes are incubated together are quite different as well, with Vesi-Dye being much more optimal and essentially giving a cleaner signal. So now thinking about dye transfer. So this was a nice experiment done using liposomes. So here we have liposomes that were labelled with DID or DIO, so two separate samples, and then mixed together at different ratios. And in an ideal scenario, you don't want to see dye transfer, but you can see that there are actually dual positive events in this middle quadrant here, starting to be detected when different ratios of these populations are mixed. And so that's giving the indication that actually this dye is transferring between those two samples when they're mixed together. In contrast, you can see that this doesn't occur with Vesi-Dye. So same principle here, two liposome populations separately labelled with different Vesi-Dyes that excite and emit in different ranges, and then when mixed together at different ratios, there's no significant population in that middle quadrant, showing that there's no dye transfer. So I just wanted to highlight that because it's so essential that we're not getting dye transfer when we're thinking about cellular uptake assays and also biodistribution in vivo tracking studies, so that we have confidence that that fluorescent signal is coming from the EV sample. So I wanted to make the webinar a bit practical as well. So I'm not going to go through a protocol, obviously, but I do want to give just some practical advice on where to start and some important considerations if you're thinking about trying out Vesi-Dye. So the first thing I'd recommend is obviously checking that your instrument is compatible with one of the dyes, one or several. We can help with this if you're unsure, or if you have several instruments in the lab that you want to obviously pick a dye that's compatible across all of them. Ideally, then we can help select the best one for your setup. More than happy to do that, not a problem. In terms of preparing the dye, we recommend sonicating if you have access to a sonicator. If you don't worry, it's just a recommendation to ensure a thorough dissolution of the dye. If not, just vortex for quite a few minutes, and spin down the dye before every use and just make sure it's properly dissolved. It's water soluble, as I said, so you can dissolve it in water, obviously, or PBS. It works well as well. And yeah, so it says in the protocol about sonication, but it's not absolutely essential if you don't have access to a sonicator. I think this is the most important point. So in terms of a recommended starting point, we suggest starting with 1 micromolar of dye for 1 × 10⁻⁻10 particles per mil. for a 1 hour incubation. But I do want to emphasize this is very much just a recommended starting point, and I would encourage you to titrate around this depending on your application. Consider both the EV concentration of your sample and then the dye molecule ratio, and try to maintain that ratio. So if you've got a sample that's more concentrated than that, we'd definitely recommend increasing the dye, and less you could decrease the dye, if that makes sense. So yeah, definitely titrate depending on your own setup. Each Vesi-Dye has its own brightness, so you need to titrate and optimize the labeling concentration for each dye. And also you could play around with incubation times as well. So there's a few things to sort of tinker around with if you're first getting started with the dye. And then recommended controls, we recommend a buffer only control. Check that your diluent is clean and it should be filtered. An EV only control is often useful for gating, and then a dye only control. So obviously we know that in our hands it doesn't aggregate and it doesn't fluoresce just in solution, but it's a great control to have there just so you have confidence that the fluorescent signal you're seeing is truly associated with your EVs. So once you've labelled the EVs, it's important then to remove the excess dye, and that's true for antibodies as well, or any fluorescent probe that you're working with. And so there's various methods that researchers use to go about doing this. So ultracentrifugation, they're all similar to methods of EV isolation in general. So ultracentrifugation, there's various pros and cons to each of these methods, but with UC, it can concentrate the samples, which can be quite beneficial. You don't really need to buy any additional consumables once you've got the ultracentrifuge in the lab, and lots of EV labs now do have them. But obviously it's very time consuming, and really I'd say the main difficulty with it, in my experience, is resuspending that pellet. It leads to significant particle loss, and it can become quite variable between samples and between days of doing it. So that's quite a challenge. There's also potential for issues with EV aggregation at that high speed spin, and obviously, yeah, it does require an ultracentrifuge. Secondly, ultrafiltration is quite often used to concentrate the samples as well as remove excess dye. It's a nice simple workflow. You can load up the centrifuge with multiple tubes at once, so it's quite scalable and it can concentrate the sample, but there is quite significant particle loss from EVs sticking to the membranes, and also depending on what kind of sample you're working with, particularly if it's with a biofluid, it might not really be possible to use ultrafiltration. You might get a lot of filter clogging happening. And then finally, size exclusion chromatography is much gentler on the EVs and gives good purity, but with that comes often quite significant dilution of the sample, between 3 and 5 times typically, and also it's quite time consuming having to collect multiple fractions. So pros and cons to all of these, and that's where Vesi-SEC comes in. So I just want to introduce Vesi-SEC, which is based on the same general principle as size exclusion chromatography, but it's formatted as a spin column. So instead of gravity flow and fraction collection, the sample's simply loaded on top of the spin column and centrifuged for 1 minute at 1000 G, eluting your purified EVs in 1 minute. And so it keeps all the benefits of SEC. It's gentle, gives you efficient separation from excess dye and antibody, etc. But it removes some of those practical limitations, especially the time it takes to process. And crucially, it's non-dilutive, so you actually maintain the concentration of EV particles before and after processing with Vesi-SEC. A van has just parked up outside, so I hope you can't hear that. Apologies if that's very loud. So in terms of performance, we're getting more than 90% particle recovery. So unlike ultracentrifugation or ultrafiltration, where you're prone to quite significant sample loss, we're getting high particle recovery with the Vesi-SEC. and there are 2 different formats depending on the sample volumes that you're working with. I suppose for this topic of webinar, most people labeling EVs are generally working with the smaller volumes, so the micro model, which is for 50 to 100 microliter sample volumes, but the MIDI works exactly the same, so there's no difference in terms of performance or how it works. So both are suitable, and the main message here is that Vesi-SEC is intended to make SEC more practical, more routine, more scalable. You can fill up your centrifuge with all of these spin columns quite easily, and in 1 minute elute your purified EVs. And bear in mind, they're also useful for EV isolation in general, not just removal of free dye and antibody. But for today, I'm going to be talking about them in this context. So I might just close the window because I think it might be loud, so apologies if it is. There we go. I think it's the bin then. Hopefully that's better. Right. So when we fluorescently label particles, we often add a dye or antibody in excess to ensure that the labeling reaction is efficient. But that excess dye then becomes a problem, as free dye or unbound antibody can contribute to fluorescence background. And I'm going to show an example of this shortly. The issue with elevated background noise is when we're measuring EVs, we're often really pushing the limits of detection of various instruments to detect those small, weak signals that EVs emit in terms of light scatter and fluorescent signal. So any kind of elevation in the background noise can actually mask those EV signals. So here is a testimonial that I did that before I was at the company during my PhD a couple of years ago, comparing measurements of BODIPI labelled EVs with a cleanup step via Vesi-SEC or a cleanup step via the QEV by ISON, or no cleanup step. So you can see here quite clearly actually that the baseline noise is elevated, whereas after the cleanup steps for both QEV and Vesi-SEC it's dropped. And essentially you can see then in these populations without a cleanup step we're only getting about 54% of the EVs detected, whereas with the cleanup step we now are able to reveal the previously hidden population that was masked by that elevated background noise, and so now we're getting around 68% of those particles being detected. So that's a whole chunk of your EV population that was previously hidden, but actually with having an optimized free dye removal protocol, you can then reveal that additional population. So the overall benefits of Vesi-SEC is it's much quicker than conventional size exclusion, and also it's non-dilutive, so you maintain your sample concentration in just a 1 minute spin. A similar experiment was done here, this time with an anti-CD63 antibody. And again, you can see that before a cleanup step, you've got a much smaller population of CD63 positive particles being detected, and post, you've got almost double here revealed. And that's because of that drop in background noise. And bear in mind, whilst I'm focusing on labelled EVs with Vesi-Dye and then removing free dye with Vesi-SEC, this workflow in general is very much compatible with dual labelling with antibodies, for example, or building multiplex panels using Vesi-Dye as a total EV population marker. And then you could look at a subpopulation of interest with an antibody, for example, and then use Vesi-SEC to clean up. So overall, hopefully you can visualize really how quick and quite simple this workflow is when we bring those 2 solutions together. And the end result is fluorescently labelled EVs with free dye removed and the EV concentration maintained. And Vesi-Dye overcomes a lot of the issues encountered with commonly used lipophilic dyes, making it not only much easier to work with, but also giving you greater confidence that the fluorescent signal you're seeing is EV associated signal. And yet Vesi-Dye and Vesi-SEC used in combination provides an efficient and reliable workflow ready for downstream applications. So I'm just coming back to this schematic because now I'm going to talk through some of the different applications of Vesi-Dye after optimized labeling with examples of users' work that has been recently published. And really, I want to demonstrate the different ways that the dye can be used and the specific value it can bring to your experiments. And a quick disclaimer before I start going through these, I'm not going to be going through the whole paper. I'm very much just taking a snippet of what is a much larger body of work. And I just want to highlight how specifically the Vesi-Dye has been used in a specific context and the information gained from it. But all of the links to these papers are on the slides, so if you do want to read them in detail, you can. So this first study, the aim of this was to determine whether EVs can be isolated from raw sheep milk and whether those EVs modulate inflammatory responses to human monocytes and also macrophages. So what they did was the authors isolated both large and small EVs from raw sheep milk by differential ultracentrifugation. And the isolated particles were labelled with Vesi-Dye and then analysed by conventional flow cytometry to identify the lipid membrane containing particles and then also determine EV abundance. And then they investigated the effects of those EVs on inflammatory profiles of monocytes and macrophages, looking at reactive oxygen species and also chemokine secretion. And what the Vesi-Dye allowed them to measure was identification of lipid bilayer containing particles by flow cytometry, giving confirmation of successful isolation of milk-derived EV populations, and then using that positive gating, which you can see here. So using the fluorescence positive gating, the authors then demonstrated the presence of both large and small EV populations in the sheep's milk. And functionally, they were then able to pull out that they have different modulatory responses in human immune cells. So they also contain various inflammation-associated microRNAs as part of the cargo. So they were starting to separate out functionally distinct populations of EVs based on size by labelling with the Vesi-Dye. And then similar to this previous study, Vesi-Dye 600 was used to confirm the presence again of intact lipid membrane-bound EVs by flow cytometry. And this study was looking at the regenerative properties of umbilical cord and adipose-derived MSC EVs for skin rejuvenation. So what they did is isolated and characterized the EVs before then looking at their effects on skin aging models. And the take-home message overall was that Vesi-Dye enabled a rapid and simple method to confirm the presence of EVs. So yes, fluorescence-based confirmation of lipid membrane-containing EVs across a diverse sample type and applications. And I'm going to continue that now, looking at fluorescence NTA. So this isn't actually published data, this is just data that we have collected via our collaborators. But another useful application of Vesi-Dye is improving specificity in fluorescence NTA. With conventional NTA, light scatter alone can't separate out EV particles from background events or contaminants. So for example, if you've got nanoscale bubbles in there or salt crystals, all of these measurements might then affect the reported particle concentration and size. And that's the same for techniques such as resistive pulse sensing as well. But by labelling the EVs with the Vesi-Dye, or in this case liposomes with the Vesi-Dye 520 or 800 on the Nanosite Pro, and then the Vesi-Dye 490 on the ZetaFew, you can then restrict the analysis to just the fluorescence positive events, giving greater confidence that the reported size and concentration data relate specifically to the EV sample. So now thinking about Vesi-Dye labelled EVs for functional studies. So I'm first going to show quite a recent study actually that was published in JEV, which actually used both the Vesi-SEC and Vesi-Dye workflow that I talked through earlier. So this is a really nice example of this workflow coming together. So overall they were investigating whether EVs contribute to inflammatory changes in COVID-19 by altering the behaviour of phagocytes, which are immune cells involved in repair. And the authors compared EVs isolated from COVID-19 patients and healthy controls. And what they did was isolate EVs from bronchial aspirated fluid, so it's the acronym being BAF, from COVID-19 patients, and then characterised the cargo of those EVs. And they found that the COVID-derived EVs had an altered composition, including depleted microRNAs and an increase in these damage-associated molecular patterns. And these appeared to reprogram the monocytes towards a senescent phenotype and changed their metabolism and also cytokine responses. So what Vesi-Dye allowed them to do was, given this apparent link between the change in EV composition and then phagocyte behaviour, they could then determine whether actually the way that the EVs interact with the monocytes was potentially altered. So they labelled these BAF-derived EVs with Vesi-Dye, removed the excess dye with the Vesi-SEC column, and then incubated the labelled EVs with healthy donor monocytes at a physiologically relevant ratio, which in their case was 1,000 EVs per monocyte for 1 hour. And what they saw was that Vesi-Dye labelling enabled them to visualise and quantify the EV uptake by the monocytes. They actually found that EVs derived from COVID bronchial fluid showed a significantly reduced interaction with the monocytes compared with EVs from healthy bronchial fluid. But interestingly, no difference was observed when plasma-derived EVs were compared. And then you can see on the right in panel B, they also did a series of Z stacks to confirm that the EVs were being internalised by the cells. So overall, Vesi-Dye essentially enabled them to directly visualise the altered EV to monocyte interactions in COVID-19, and then this kind of set up gave important mechanistic insight into how those bronchial EVs might be driving inflammation in COVID patients. So really, really interesting. Another example of this, which is somewhat similar in terms of looking at uptake, cellular uptake by confocal microscopy. This study was looking at engineered milk extracellular vesicles that were hybridized with, if that's the right term, they were fused to form hybrid particles with LMPs or lipid nanoparticles. And the application of this was oral delivery of siRNA. And so what they wanted to do was generate these hybrids by fusing milk-derived EVs that had been labelled with the Vesi-Dye with lipid nanoparticles that contained that siRNA as a method of loading EVs with the siRNA. So then essentially the Vesi-Dye enabled them to visualise the EV membrane within the hybrid nanoparticles. They did this by some super resolution microscopy as well. And then they also wanted to confirm cellular uptake of these hybrid particles by intestinal epithelial cells, which is what is shown here. So combining with, they did various pharmacological inhibition studies. They also were able to demonstrate that the hybrid particles actually were taken up by distinct pathways, suggesting that the engineered EVs have an altered behaviour compared to just the LMPs and just the EVs on their own. So overall, Vesi-Dye enabled the visual confirmation of uptake of these hybrid particles, supporting the development of a novel oral RNA delivery platform with enhanced intestinal transport. And I just really like this image here, which is why I kind of blew it up a bit, so you can see, hopefully, you can see those little red dots that represent the labelled hybrid particles with Vesi-Dye. So finally, I just wanted to show this one last example because they're all quite different in a way. So I think this is a really nice example of in vivo biodistribution. So this study was looking at the function of red blood cell derived EVs. And the authors were interested in where they localize in vivo and which cell types preferentially take them up. So again, this was the Vesi-Dye was used to track essentially where these EVs are going. So the red blood cell EVs were fluorescently labelled with Vesi-Dye 490 in this instance and injected intravenously into mice. And then after 8 hours, the organs were collected, harvested and analysed by flow cytometry and also Confocal. And then they also incubated the Vesi-Dye labelled EVs with human PBMCs as well to just see which immune cell populations preferentially take up the EVs. So the stability and the brightness of the Vesi-Dye enabled that in vivo tracking and also the identification of the target cell populations. So this was a critical first step really in determining which cells interact with red blood cell EVs, and then this guided their subsequent mechanistic studies of EV uptake and intracellular trafficking. And you can see here that essentially what they saw was the liver and spleen seemed to preferentially take up the EVs, specifically by resident macrophages. And yes, in vitro, they showed that CD14 positive monocytes showed the greatest uptake amongst the PBMC populations, highlighting that macrophages and monocytes appeared to be the key target cells of the red blood cell EVs. So overall, Vesi-Dye essentially enabled a robust in vivo and in vitro EV tracking. They also looked, you can see these images up here are actually histological sections. So you can see how stable the dye actually is, having been processed that way. So yeah, overall the Vesi-Dye enabled in vivo and in vitro tracking of EVs, allowing the authors to identify macrophages and monocytes as the major target cell of red blood cell EVs, and this provided the foundation for their mechanistic studies. So I've shown a few different examples that have been published of how researchers have used the Vesi-Dye in their experiments. But there are many more published examples I haven't gone through, and we're increasingly seeing users apply the dye in different ways in different types of experiments. So it's very much just scratching the surface of what the Vesi-Dye can be used for and the information that it can tell you about EV biology. And I've got some sort of ideas that I wrote down here that might be relevant for people of other things that you could do with Vesi-Dye. So yeah, in conclusion, hopefully I've sort of shown you that stable EV labelling is really important for strengthening EV analytics and functional assays. Faster EV purification workflows increase the efficiency and scalability. And also Vesi-Dye in combination with Vesi-SEC provides an optimized workflow for reliable fluorescent EV staining and analysis. And across all of these areas, our goal is the same. We want to help preserve sample quality and also improve the confidence in the final data. So I think I whizzed through that in hindsight. Oh no, gosh, the time's gone quickly. Okay, cool. We've still got plenty of time for questions though. So yeah, I hope that was a good overview of kind of what Vesi-Dye can do and how it can be used in combination with Vesi-SEC as a complementary optimized workflow. And if you've got any questions about the dye, or Vesi-SEC, let me know now and we can discuss them. So I'll stop sharing my screen for now. Okay, let me just have a minute checking through the comments. Right. Oh, it's not loud. Okay, I'm glad that it wasn't loud. Sorry, I couldn't hear anything, so I was worried. Okay, right. So we've got questions in the chat and the Q&A. So I'm just give me a second to go through those. Oh yes, lots of questions. Okay. Oh, sorry if the graphs were a bit too small. I mean, that one plot is particularly bad for that. But I'm more than happy to send any of the data that I've showed after this. So if there's anything in particular you want me to send, do let me know, and that's fine. Okay, so Oliver, is it essential to re-NTA/BCA the samples after using Vesi-SEC before going on to downstream experiments? No, it's not essential. I think what I would do in your scenario is I would check that just for my own knowledge and insurance that the same sort of number of EVs, or depending what you're looking at, I guess, particles and protein. I would personally measure them just so I knew for my own confidence that the Vesi-SEC is doing what we expect. But yeah, it's not essential. The Vesi-SEC simply is a size exclusion chromatography column. It's got a 35 nanometer cutoff, so anything below that is retained in the resin, and anything above that is eluted in that 1 minute spin. So it works very simply like that. So can I stain EVs directly in biofluids like serum or plasma, or only isolated EVs? I would strongly recommend isolating the EVs first. In biofluids such as serum and plasma, you're obviously going to have lots of lipoproteins in there, and the dye probably isn't specific enough to not label those lipoproteins. So I think very important to try and remove lipoprotein contamination as much as possible so that you get a clean EV sample for staining, essentially. So yeah, we would recommend for optimized staining, you really want to isolate the EVs from those biofluids, ideally. I think I've just answered this question by Claire. Yeah, if you have a heterogeneous mixture of EVs, lots of different sizes, and something like LDLs, does Vesi-Dye also localize to those particles as well, or is it EV specific? So it intercalates the bilayer of membranes, but I think it's not specific enough that it wouldn't also bind to some degree other lipids or lipid particles that are present in the sample. So yeah, I think we can't claim that it's fully 100% EV specific when there's lipoprotein present. Can you explain in more detail why Vesi-Dye doesn't create aggregates on micelles? So it will be down to the unique chemistry of the dye, which is an Acarella technology that's undergoing patent filing. So I don't know the exact chemistry of the dye. It's confidential. So sorry. So hi Oksana. Thanks, Oksana. I would like to check if Vesi-Dyes are stable when injected in vivo and the animal sections processed via wax embedding. So in the last example I showed, actually, the Vesi-Dye was detected they weren't wax embedded, but they were frozen cryosections of tissue. And that is after injection in vivo intravenously into mice. Those organs were then harvested and processed by histology by freezing. Is it snap freezing? I can check the exact method, but yeah, and the dye was still visible in those sections along with other markers that they checked for. So yeah, it is stable enough to do histology essentially. I don't know if we have an example of exactly wax embedding, but I could look into that, and I'm sure they would be stable enough if they're fine for cryosectioning. Yes, Mo, the slides will, the webinar recording will be available, and we'll circulate that probably early next week. So yeah, that's absolutely fine. So if there are disintegrated broken EVs in the sample and they aggregate, would Vesi-Dye stain them and assume they are intact EVs? So if there's fragments of membrane, the dye will bind to that. Doesn't necessarily mean that they appear like intact EVs. It depends on kind of the method you're using to measure the EVs. But yeah, it will bind to them and it will then fluoresce. So that's important to remove those if possible. Yeah, Claire, I've just seen your comment about, yes, they can be the same size. So they are often present after ultracentrifugation. Yeah, so we do have actually relatively new workflow that we launched earlier this year that is for selectively precipitating, can't say that word, out lipoproteins from plasma samples whilst maintaining intact EVs. So if that's something that might be of interest, do let me know, and I can give you more information on that. But yeah, they are the same size. HDL is obviously smaller and will be retained in the resin of the Vesi-SEC column, but the LDL will elute through with anything that's the same size, as EVs or anything above 35 nanometers. So yeah. Okay, cool. Yeah, I'll send you that information. Should the Vesi-Dye need to be pre-stained with cells before EV conditioning? So the protocol that I mentioned earlier is about incubating and labeling EVs post isolation from cells, as opposed to labeling cells to then acquire fluorescently labeled EVs, which I'm not sure would work, and we certainly haven't tested. So yeah, the protocol that I described was simply for once you've isolated the EVs to then label them with the dye. If I need to double stain functional mitochondrial marker, should I do it before or after the Vesi-Dye or together? It might be a case of trying all three conditions and seeing what works optimally, because I don't know enough about mitochondrial marker dyes like MitoTracker. I mean, ideally, you wouldn't see any difference between those three conditions. So that would be something to test. Right, I'll switch over to the Q&A because I think there's loads in there as well. This is great. Thank you for all your questions. Do you also have the Vesi-Dyes for targeting bacterial EVs in a mixed population? So the Vesi-Dyes, they'll bind anything with a lipid bilayer, basically. So they will certainly stain and bacterial EVs. By mixed population, I'm not quite sure what you mean, but yeah, they're not specific to bacterial, for example, but they will just label anything with a lipid bilayer. So yeah, they'll certainly work for bacterial EVs if they have a lipid bilayer, which they do. Does the Vesi-SEC or Vesi-Safe help in prevention or elimination of EV aggregates? So the Vesi-Safe will prevent EVs sticking to plastics. I don't know that they prevent EV aggregation. The Vesi-SEC certainly won't cause EV aggregation, like something like ultracentrifugation, which obviously the force of that can cause sort of aggregates and fusing of EVs and also damage to the EVs. So yeah, the Vesi-Safe in comparison to that is much gentler at just a 1000 G spin. But so actually elimination of EV aggregates, depending on the size of the aggregates, the Vesi-SEC won't eliminate them because it's anything above that 35 nanometer cutoff. So they would come through with the vesicles. So you might, if you're having a problem with EV aggregation, maybe you'd need some sort of pre-clearing step before Vesi-SEC, maybe. Yes, we can share the webinar after the session. We'll be doing that early next week. have you used these columns for small volume / low concentration samples? Yes. My samples come out indistinguishable from PBS on nanoflow cytometry when using columns in these cases. So I used, if you're talking about Vesi-SEC, I used Vesi-SEC a lot during my PhD and postdoc. And I found them really useful for low concentrated samples. And that's really kind of where I started using them quite routinely. Because they're non-dilutive, they retain your EV sample. So you're not losing anything, and it makes it very easy then to apply quite a lot of antibody or dye and then just wash it out without diluting the sample, and then you can measure it for nanoflow quite nicely. So I found that they work really well for that, and the lower sample volume cutoff is 50 microliters for the micro model. So yeah, that could certainly work, and I'm happy to chat more about that, Max. I don't actually know how bright the Vesi-Dye is compared to PE off the top of my head, but I can find out and I'll let you know. I'm going to note that down so I don't forget. Does the dye integration have any impact on the uptake dynamics or functions of the particles in vivo? Not. Well, it's not something we've obviously personally tested, but not that we're aware of. And there's many publications now that are using these dyes for uptake and for biodistribution. The dye doesn't affect the size of the particles, it doesn't cause any kind of swelling, it doesn't cause any aggregation of the particles. So yeah, it shouldn't, but obviously that would probably be worth testing in your context as well. Hi Josh, thank you. I have found that antibody alone and SEC can often result in antibody coming through when measured by nanoscale flow cytometry. Is this something you've seen before? So it will depend what concentration you're using. This is something we've talked about before. So often, I guess you would match the concentration between your EV sample, whatever you're incubating, with that, to then mix with buffer to put on the SEC column. But if the epitope's really highly expressed on the EVs, you might find that you're getting some coming through. It depends on kind of the threshold. I can talk more about that with you and figure that out. It's a bit of a tricky one, but yeah, overall it shouldn't be coming through. So it's obviously oversaturating the column, the amount of antibody there. But yeah, it should enter the resin and be retained if it's at the right sort of concentration. So will the Vesi-Dye have an impact on the functionality of the EVs? Not in theory, but yeah, I think obviously functionality of EVs is so broad, it would be worth checking whatever your research question and context is. Have you checked background noise from the dyes on nanoflow cytometry? Yes, yep. So I did show a plot earlier of just the dye mixed with buffer, and you see that there's virtually no background there. So yeah, I can provide more data, Tatiana, if that's of interest. Is the Vesi-SEC column designed for single use, or can it be reused like the eyes-on column? We recommend single use. We have had users sort of test reusing the columns. Basically, if you want to reuse the columns, you'll have to test that in-house for your own application, depending on your own EV samples, because it will vary so much between different users. But yeah, we certainly recommend single use for the Vesi-SEC columns. Oh, interesting. Does the lipid composition of EV membranes affect the Vesi-Dye labeling, and have you tested on EVs from different cell types? So I don't think so. I like that question because that just makes me think about how the chemistry of the dye is working, which we don't know what the chemistry of the dye is yet. But it's certainly been tested on a wide range of different EVs from different cell sources, and also cells themselves, many different cell types can be, stained with these dyes as well. Obviously, I focus the talk on EVs, but yeah, so there's a lot of data now, published data out there using these dyes on various different types of EVs and cells. So we don't think so, but Yeah. Do you have any products that efficiently separate viruses from EVs? No, unfortunately not. They, as far as I'm aware, I'm not a virologist, but as far as I'm aware, viruses very much overlap in size with EVs. So that is a difficulty in terms of size-based separation. And the Vesi-SEC is a size-based separation method. On a similar note, does the Vesi-Dye label viruses such as lentiviral vectors as well as EVs? Yes, it does. Yeah. Will Vesi-SEC work for a range of different starting materials? Yes. So yeah, again, I focused the context of the talk on just EVs, but actually it will work for a wide range of different nanoparticles. So for example, liposomes, lipid nanoparticles, all of these can be used to clean up free dye or free antibody or whatever. Have you conducted a detergent treatment to confirm EV labeling was successful? And have you tested dyes on bacterial EVs? We might have. I'm sure we'll have data for bacterial EVs. Yeah. Let me write that down because that'll be another thing I can find for you, Tatiana. Yeah, I'm sure there must be. I can't think off the top of my head, but I'm sure there would be by now. And have you conducted detergent treatment to confirm EV labeling was a success? I'm not sure which part of the talk you're referring to for that, so I'm happy to chat about more. Obviously, yeah, detergent control is a great way to confirm that you have EVs in a sample by then eliminating them, and is very much recommended by the flow cytometry guidelines for EVs. How well did the dyes work together, co-staining the EVs? So if you wanted to use two colours together on the same EV, I'm not sure I know the answer to that question, actually. So I will get back to you on that, Diana, because I don't think we've tested that. And I'm trying to think if I've ever seen any data that's actually combined the two dyes. I mean, there's no reason why it wouldn't work, I suppose. Yeah, I don't know. I'll get back to you on that, Diana. Thank you. Can Vesi-SEC help in the removal of EV aggregates along with unbound dye removal? So EV aggregates will be bigger than the EVs themselves. So no, Vesi-SEC simply excludes anything below 35 nanometers, and anything above that will elute through. Does Vesi-Dye stain lipoproteins? I think it's likely that it probably would to some degree. I'm not sure how efficiently, but I think it's likely it would label lipoproteins. Yes, the prices are listed on our website, Diana. If you go to the shop section of the menu, all the prices are there, but equally happy to send you a quote for anything that you need. Does Vesi-SEC perform well in a microcentrifuge? Yes, absolutely. That's what it's designed for, actually. So our first micro model was the first one we brought out, and that is designed for a microcentrifuge. And then we've also got the MIDI model, which goes up to a mil sample volume and uses 15 mil tubes. Would the Vesi-SEC work for large EVs as well? Yes, yeah. So like I said, anything above 35 nanometers essentially is eluted. There might be a bit of, say, anything above, I suppose, like you've got to get rid of the cells, you've got to get rid of sort of large micro debris. I'm not sure how well it would perform if you didn't do that. So it'd probably clog up the resin a little bit, but otherwise large EVs, anything below 1,000 nanometers should work well. What is the stability of the aqueous solution? I'm assuming you're referring to the dye. So I think it can be stored in the freezer for up to a year. I will check that. I can probably find that out now, actually. I'll just answer the very last question from you, Colby. Have you looked at the portion of EVs within a sample that are labelled by the Vesi dyes, and are there any EVs that don't get labelled? Not that we've seen, but it would be interesting, similar to that earlier question about specific sort of lipidomic composition of EVs, whether that affects the staining efficiency. That would be really interesting to find out. In terms of stability of the aqueous solution. Let me find that out now. Or maybe I can get back to you in a minute, because that will be in the protocol, which is also downloadable from the website actually, but I'm pretty sure it's 12 months. Obviously that's for the stock solution. I suppose if you're diluting more to then label your EVs, we'd probably recommend just using that as soon as possible, but for the recommended stock solution, that can be stored for about a year, I think. So I will find that for you now. Okay, I will send you that afterward because I've got too many documents open and I'm getting lost in them. Are there any more questions? Let me flick back over to the comments. Maria, wish I could stay longer, but the centrifuge is done. Fair enough. Thank you so much for giving up your time listening to me, and thank you for all your questions. I don't think I can see. Oh, hold on. Heng's just sent one. For centrifuge in the column, should we always stick to 1000 G 1 minute, or is it more important to keep the same condition as washing the PBS? Also, loading or labeling EVs, the cargo or the dye size has to be 35 nanometers. So we've actually got slightly different spin speeds for the wash steps between the micro Vesi-SEC model and the MIDI. So the MIDI has a slower 500 G spin step for removing the storage buffer before loading your sample. I don't want to confuse people because both of them work the same and both of them use 1000 G spin for eluting your actual sample. It's just there's 2 steps before to wash the column. So whatever it says in the protocol, basically follow that depending on which model you're using. For the micro model, it's always 1000 G. For the MIDI, it's a slower speed to remove the storage buffer and wash with PBS before then loading your EV sample and eluting at 1000 G. And then in terms of the 35 nanometer cutoff, all I'm saying is basically you load your sample onto your EV sample onto the column, and anything below 35 nanometers will be retained in the resin. And so in our context of today, I'm talking about dye molecules and antibodies. Could also be soluble proteins if you're doing EV isolation. It depends, yeah, what you're doing. Some of the text on the website isn't appearing. Ah, thank you. I can only see the images on the product pages. Okay, cool. Thank you for that feedback, Colby. I'll have a look into that. And thanks, Oksana. No worries. Okay, just flicking back to Q&A, but otherwise, not seeing anything else. I don't think. Yes, so I think that's it from everyone. You're welcome, Heng. So yeah, I think we'll wrap up there then. Yeah, we will share the recorded version probably next week, early next week. So that will go out to everyone that registered to the event. So you'll get that in an email. And yeah, I hope that was useful, and I hope I didn't whiz through things too quickly. And yeah, thank you for your time. Great, thank you. So yeah, do get in touch if you have any questions or you want me to send anything further. More than happy to do that. Thank you. Thanks, everyone.